To use the PicTar web interface to identify candidate genes of microRNAs, you first need to navigate to the PicTar website, which you can find in the Lab 3 folder on Brightspace and in the pre-lab document. Once on the PicTar web page, you'll leave most of these settings as they are. In the microRNA ID tab here, you'll select your microRNA of interest. So in this case, I'm going to select microRNA 23B. Once you've selected your microRNA, you just need to click on this Search for Targets of a microRNA button at the bottom of the page. This will take you to a new page with the output of PicTar's predictions. Now there's a lot of information on this page, but you only really need to focus on a couple of key points. First of all, on this page, PicTar has listed potential target genes for microRNA 23B ranked based on their PicTar score. And that PicTar score is just in this column right here. The PicTar score is an estimate of the strength of the prediction. So from this list, you can select any gene that you might be interested in as your candidate gene. However, before picking your gene, you might want to get a little bit more information about it. So let's say, for example, I was interested in this top gene here, a transcript variant of the SEMA6D gene. If I wanted to get more information about this gene, I would click on the link right over here. This is actually the extension number for the gene. The extension number is a unique identifier that's been given to the DNA sequence of the gene, and it can be used to record and track different versions of the sequence through various sources and over time. So by clicking on this link, it will take me to the GenBank database, where you'll find a whole bunch of details about the gene. Again, there's a lot of information on this page, and we encourage you to explore it. Note, for example, right here, you see the extension number for the gene, and this is the same as the number on the link you clicked to navigate to this page. I'd like to draw your attention to a few key points. First of all, at the very top here, you'll find some important information about your gene. So you'll see the size of the gene. So in this case, it's 6,109 base pairs long. You'll also find the type of sequence. So in this case, an mRNA. And it also specifies that the sequence is linear. So remember, in some sequences, such as those from viruses, it will actually be circular. Just below, you'll find the source organism for the gene. So your genes should also be for Homo sapiens, because we're working with human cell lines. And below that, you'll find a list of references or primary publications that were responsible for all of the information on this page. If you look at the side column on the right, you'll also find a list of primary research articles about the gene you've selected. Above this section, if you look just a little bit higher, you'll find the Pick Primers link. Now this will be really useful in the next lab, or in the next lab when you're going to be designing primers for your gene. Now, most importantly for your lab, you'll find at the top left of the page a FASTA link right here. Now, FASTA refers to a text-based file format that's used to represent sequence data. If you click on this link, you will find the DNA sequence for your gene. Here it is right here. You will need this FASTA sequence later on to design your qPCR primers. There are a couple of ways you can save this information or save this FASTA sequence data, but the easiest for our purposes is simply to copy and paste it from the web page into a Word document. Make sure you copy the entire gene right to the very end. Make sure that you record your gene name and the extension number for your gene in your lab manual, and that you save your FASTA sequence.